of the genetic material. One is that is the genetic material means DNA, and next cutting of DNA at specific locations, and the next amplification of gene of interest using PCR. So insertion of rDNA into the host cell, and the fifth one is the obtaining the foreign gene product. So next one, sixth one is the downstream processing. So these are the very important steps for the understanding the DNA technology process. So sometimes in a NEET examination also identify the steps of the DNA technology which is correct or which is not correct. So that's why these six steps are you remember it and by heart also. So first one, what is that? First, isolation of the genetic material DNA, and next one, cutting of DNA at specific locations, and the next one, amplification of gene of interest using PCR, and insertion of rDNA into the host cell, and the next one, obtaining the foreign gene product, and next one, downstream processing. So these six steps are very important. Okay, now here, the process of recombinant DNA technology, the first one is, well, the first step is what is that? Isolation of the genetic material DNA. The genetic material is what is that? DNA. So here, you are observing the removal of first one, the removal of plasmid or genomic DNA from cells is a termed as isolation. Isolation means what is that? So from the so from the cells which you want, that can be removed by the process. The process is called as what is that? Isolation. Isolation means what is that? Separation. So or remove. So what is that? The removal of plasmid or genomic DNA from cells is termed means what is that? Isolation. So next one you are observing. These can be achieved by treating the bacterial cells or plant or animal tissues with enzyme. How it is possible? For example, the plasmid is inside the cells, so that's why first is necessary to the cut or the open the first the outermost layer. Outermost layers means what is that? Which is considered as what is that? Cell wall. So that's why first one outermost layer is cutting. Then after we can get the which you want, so means genomic DNA or plasmid. So here you clearly observe that this can be achieved by treating the bacterial cells or plant or from the anyone, so bacterial cell or plant cell or animal tissues with enzymes, so such as, so these are very important you are observing. Lysozymes. So these lysozymes, MJMEs. So what is that? React on the bacteria and the cell wall, and open the cell wall and digest the cell wall. By the enzyme name is what is that? Lysozymes. What is that? That enzyme name is what is that? Lysozymes. And next one, in a plant cell, generally the plant cell wall is made up with the what is that? Cellulose, hemicellulose, pectin, etc. Substances are involved into the made of the cell wall. So that's why here you are observing generally the plant cell wall is made up with the cellulose. So that's why the cell wall is destroyed or digested by the a particular or specific enzyme that is called as what is that? The cell wall is made up with the cellulose. So you can add the what is that? Ase. The enzyme end with the what is that? Ase. That's why cellulose plus ase. So that's why the enzyme name is called as what is that? Cellulase. So the plant cell is digested by the which enzyme? Cellulase. And the next fungus. Generally the fungi cell wall is made up with the chitin. So the chitin. So that's why the chitin is digested by the enzyme is what is that? Ase. So chitinase. What is that? That enzyme is what is that? Chitinase. And the next one you are observing these enzymes are, first one what is that? Isolation of the genetic material DNA means what is that? Isolation means remove the plasmid or genomic DNA from the cell. So how it is possible to the remove the plasmid? So first it is covered by the what is that? Any membrane. So that membrane after the cut the or digested the outermost layer, then after the plasmid or genomic DNA can be exposed. So for the digestion purpose or the open purpose, we can treat the some specific enzymes like here lysozymes means bacterial cell wall is digested by the or dissolved by the enzyme is what is that lysozymes and plant cell. So generally the plant cell wall is made up with the cellulose hemicellulose. That's why the cell wall is digested by what are dissolved by the what is that the reaction of the cellulase. And fungus or fungi, generally the fungi cell wall is made up with the chitin. So that's why the fungus or fungi cell wall is treated with the chitinase. Automatically, the cell wall can dissolve or digested. That's time inside the plasmid or genomic DNA can be exposed or removal from that. So these process is called as what is that? So isolation. So here you are observing clearly. Genes are located on long molecules of DNA intertwined with the proteins such as the histones. Already you know the histones proteins are there. So in a, about the histones and the remaining proteins and the DNA linkers. So these are histone cores we can study in the nucleosome model. So and the if the treated with the proteins, so proteins. So for the digestion of proteins, we can use it or we can treat the with the proteases. Protein plus is proteases. Chitin plus is chitinases. Cellulose plus is cellulase. So that's why the substance you add the is automatically that enzyme name will become. So here what is that? The protein digested by the proteases. So simple you are understand. The plant cell wall is digested by the cellulase. The fungus cell wall is digested by the chitinase. Similarly, protein, histone protein that is also treated or the digested by the proteases. So the end of the point is in this. So only simple point from the cell. So removal of the plasmid, that process is called as isolation. For the isolation purpose, we can use it or we can treat it with the specific enzymes for the digesting the cell wall. Okay, that is the process of the first one. And here, for example, the RNA also be. If we want the RNA, that time also the RNA can be removed by the ribonucleases enzyme. So, for example, protein can be removed. So, for the digestion, so that's time we can use the what is just proteases. Okay, RNA is ribonucleases. Protein means proteases. Okay, simple. Chitin means chitinase. Cellulose means cellulases. So these are the enzyme names, very very important. So remember it. What is that? Ribonuclease means the RNA. Okay. Next one here you are observing other molecules can be removed by appropriate treatments and purified. Uh, mm, Automatically it can be comes out as a precipitates out after the addition of chilled ethanol. So precipitate. So ultimately, so you are observing first one what is that? After digestion the cell wall, after digestion the cell wall, then after we can what is that? Other molecules can be removed by the appropriate treatments and purified and ultimately precipitates out after the addition of chilled ethanol. When we can added on to the, so removed the plasmids, automatically you can add the chilled ethanol. We can observe the precipitate can be comes out. So you are observing this is the precipitate. So after adding the chilled ethanol, okay, this is called as what is the isolation process. Then second process you are observing what is that? Cutting of DNA at specific location. So here clearly you are observing purified DNA molecules are incubated with restriction enzymes. So then after optimal conditions are maintained for a particular enzymes. So in this, the progress of restriction enzyme digestion is checked with the help of Agarose Gel Electrophoresis. So here you are clearly observe that after precipitate means we can get the what is that purified DNA molecules, and then after it can be incubated with the some restriction enzyme. Optimal conditions are maintained for the particular enzymes. So here the progress of restriction enzyme digestion is checked with the help of agarose gel electrophoresis. So here, as the DNA is negatively charged and it moves towards the positive electrodes means anode. So this process is repeated with the vector DNA as well as. So the cut portion from the source DNA and the cut vector means what is that? The gene of interest from the source DNA and the cut vector are mixed and ligases enzyme is added. What is that? Ligase enzyme is added. And the next, these result in the preparation of recombinant DNA. And here you are observing, so by the restriction enzymes we can cut the specific places guanine adenine, you are observing guanine adenine, and here guanine adenine. So by the like Escherichia coli or ECOR1, so these restriction enzymes we can cut the specific places automatically, we can get the so specific ends. You are observing it is cutted between the where in guanine and between the adenine. Okay, so after this you are observing in these cut the where places adenine. Here also adenine between the adenine. Here you are observing ECOR is cut the between the spaces. What is that? Guanine adenosine. So different kinds of the distinction enzymes cuts the DNA strands and at different locations. So after these fragments, the by the help of the restriction enzymes, they can cut the specific places number of fragments of the DNA. Then after here you are observing here the cut the different kinds of the fragments are we can get in this process by the action of the restriction enzymes. After complete the restriction enzyme action, we can use the one technique. The technique is the a specific technique for the separation of the which are cutted by the restriction enzymes. The fragments can separated purpose. We can use the one technique that is the called as what is that? Agarose gel electrophoresis. In this you are observing this is the agarose gel electrophoresis of DNA. In this we are use so anode uh, cation and anion, we can use plus and minus charges. These are called as what is that? Electrodes. And in these we can use the you are observing buffer solution, and the we can gel. The gel means we can get the from the Phaeophyceae algae, so we can get the gel, agarose gel we can use it. And the this is the sample uses places, and the we can observe these places is as a bands like structures. So means bands means visible after the suitable treatment. After treatment we can observe the different kinds of the colors, the bands can be observed. Okay, so this is the we can use the what is the buffer solution, and the, as a user as a matrix is the agarose gel. So in this agarose gel electrophoresis you are observing, so this side is what is that? Cathode, this side is anode. So we are used to the DNA fragments, and that they can when we are gives to the electrodes to the attach to the cathode and anodes, and the different kinds of the DNA fragments can separated as the place of what is that? Small sized here. When we are gives to the attached to the electrodes, automatically the agarose gel can form the a small pores. So automatically the small pores, the large size pores are some DNA bands appeared. In the small pores places appear the small DNA appears, but these are clearly visible in the ethidium bromide treatment. After ultraviolet rays, we can observe the particular colored the bands. So we can separate these. You are observing this side is cathode side is some large size DNA bands can appear, and the anode side is very appear the very small size the DNA bands can observed. So by the after completely we can visible after complete the treatment with the ethidium bromide. Okay, so let us now we are discussing here gel electrophoresis means you simply remember it. This point also very important in it. So that's why you are clearly understood this. So agarose gel electrophoresis is a simple and the highly effective method for separating, uh, identifying and the purifying DNA fragments. So separation is carried out under an electric field applied to gel matrix. So the gel matrix means agarose, which is Agarose means a polysaccharide extracted from the seaweeds. Phaeophyce, exactly brown algae from the seaweeds, we can get the agarose. And some species also agarose can give to the rhodophyce, but very popular into the Phaeophyce, brown algae. Okay, what is that? The separation is carried out to the what is that? Carried out under an electric field applied to gel matrix. So what is that? Agarose, which is a polysaccharide. So that is the agarose is the one type of what is that polysaccharide extracted from the seaweeds. And the next, because of the negatively charged phosphates along the backbone, and the DNA molecules migrates towards the anode. So this is the very important. So because of the negatively charged phosphates along the backbones, DNA molecules migrate towards the anode. So then after you are observing under an electric field through a medium or matrix. Medium means agarose. So they separate according to the size through the pores of agarose gel. The pores can form. So next, smaller the fragment size, further it moves. The separated DNA fragments only can be seen only after staining the DNA with compound known as ethidium bromide. This chemical name is very, very important. What is that? Ethidium bromide. So what is the ethidium bromide? And followed by the exposure to the UV ultraviolet radiation as a bright orange color bands can be appeared. So the separate band of DNA are cut out from the agarose gel and extracted from the gel pieces. So this is the what is that? Here you clearly observe that ethidium bromide. Ethidium bromide is very, very important, followed by the exposure to the UV radiation as a bright orange colored bands. So in this step known as a elution, this separation process is called as what is that? Elution. This the word is very important, elution. And their purified DNA fragments are used in constructing rDNA by linking them with cloning vector. So simply you are observing the in these gel electrophoresis, you are observing what is that agarose. Agarose is observed the complete process is you gel electrophoresis is what is that ethidium bromide, and they exposed to the UV radiation as a bright orange color. So this is the very, very important. And the next one you are observing this diagram also you are observing. So here large bands are observed and smallest bands are observed. The gel can shows the small pores. So that is the very, very important agarose, which is polysaccharide and extracted from the seaweeds. Okay, now we are observing ethidium bromide. So agarose gel electrophoresis uses your observe. Estimate the size of DNA molecules. This is the first important. Estimate the what is the size of DNA molecules. And next one, analyze PCR products. Example in molecular diagnosis or genotyping. In this process, we can use the what is that agarose gel electrophoresis and the determine the quality or quantity of DNA. Okay, determine the quality or quantity of DNA. And next use is purification of DNA. So this is the very, very important for the agarose gel electrophoresis uses is first one is what is that estimate the size of DNA molecules, and next one is the analyze the PCR products, and example is what is the in molecular diagnosis or genotyping. Next uses is what is the determine the quality or quantity of DNA, and next one is the purification of DNA. So let us now insertion of isolated gene into a suitable vector. So insertion of isolated gene into a suitable vector. So isolated gene is we are separated by the uses what is that agarose gel electrophoresis method. Then after we can get the isolated gene. Then we can insert it to the a suitable vector. Already vector characteristic nature is yesterday we completed. So that's why now. So which are we are isolated from the treatment of the enzymes. Then after we can separate it by the using the method is gel electrophoresis. After that, so we can that to isolate a plasmid from the bacterial cell and subjected to centrifugation to separate the plasmid. After centrifugation, we can get the separate the plasmid, and after the cut the source DNA as well as the vector. Simple. So what is that? First cut the source DNA, means plant or animal, anyone. Then after, as well as vector DNA also cut the by the process is restriction enzymes. So then after DNA with the same restriction enzyme, the cut DNA of interest from the source, DNA and the cut vector are mixed and ligase is added. So after these result in the formation of recombinant DNA. So these are recombinants. That's why we are called as rDNA. So chimeric DNA means different kind. So one is the what is that source of DNA, source of DNA fragment added with the vector DNA. So two are different. One is the source DNA, another one is the vector DNA. So these are foreign DNA plus vector DNA. Both are the combined in the presence of the ligase enzyme, means ligation process. That's time we can get the or resulted the a new DNA. So these DNA consist of the two types of the characteristic natures. One is the foreign or the source DNA nature, and remaining half of the part shows the vector DNA. So both characteristic natures can be appeared in the recombinant DNA. So new DNA. So that is called as recombinant DNA and rDNA, and another name is also chimeric DNA. So that is the you are observing. It is also known as cDNA also. So different names are there. So smaller DNA, recombinant DNA, chimeric DNA, cDNA, all are same. So next one, here you are observing this diagram is very very important. First one, source DNA. Source DNA means our foreign DNA. It means plant or animal, anyone. So this is the vector DNA. It means what is that additional a circular replicated DNA in bacteria, that is called as what is the vector. So this is the vector, this is called as plasmid. Now you are observing the foreign DNA. It is the double helix structure. So it is can these foreign DNA can cut by the, so what is that a specific point by the restriction enzymes. Here cut the specific place with the help of the restriction enzymes, and the plasmid places also involve the restriction enzymes. The both restriction enzymes are involved for the cut the DNA fragments at the specific places. So after complete the restriction enzyme action on the foreign DNA and the vector DNA, we can get the fragments of the foreign DNA fragments as a small fragments we can get here, and similarly vector DNA also we can get the uh, some places, so fragment also. So these places here you are observing, this is the plasmid, and this is the foreign DNA fragments. Both are the, so you are observing these foreign DNA fragments can added in this place where the restriction enzymes are cut and the leave the sub gap. In these places we can add the, so which are cut the from the foreign DNA by the restriction enzymes. The piece can inserted in the gap places of the plasmid. Okay, so this process is called as what is that ligation. Because of this enzyme is called as ligase. Ligase join the foreign DNA to plasmid in these places. This process is called as what is that ligation. Okay, clear. Ligation. After this, we can get the, these are called as rDNA. So these are called as recombinant DNA, chimeric DNA, cDNA. Why? Because these is consist the what is that? Some is the plasmid nature, and another one is the what is that foreign. So both characteristic nature are there. These are called as what is that? So rDNA or cDNA, chimeric DNA. After formation of the rDNA, for the replication purpose, for the getting the number of copies purpose, so these rDNA can so inserted to the what is that a suitable so host tissue. So what is that we can selected the what is that Escherichia coli. These process are called as what is the transformation. So yeah, after inserted the rDNA, but it is not a permeable to the insert the rDNA into the bacteria cell. For the permeable purpose, we can take the another treatment. So with the calcium. So calcium treatment. After calcium treatment, the Escherichia coli bacteria can permeable to the rDNA inside the cell. So after these, they can multiply and they involve into the replication. They can produce the a large quantity of the rDNAs. So these process is called as generally what is the rDNA technology. In this you are observing. Next one, what is the amplification of gene of interest using the PCR? So PCR means you are observing PCR, polymerase chain reaction. PCR means what is that? Polymerase chain reaction. The This PCR technique was invented by the Carey Mullis in 1985. So that is the important. PCR is invented by the what is that? Carey Mullis in 1985. So what is that next one? PCR is best defined as the DNA replication. PCR polymerase, poly means number of, chain means number of, and reaction. They can produce a number of chain reactions. So by this method, so that is called multiple copies can get with the help of the PCR. So here the PCR is the best defined as the DNA replication in vitro method. In this reaction, multiple copies of the gene DNA of interest is synthesized in vitro using two sets of primers, and the enzyme is DNA polymerase. The enzyme is what is the DNA polymerase. In a PCR polymerase chain reaction involved the specific enzyme. That enzyme is called as what is the DNA polymerase. The enzyme extends the primers using the nucleotides provided in their reaction and the genomic DNA as a template. What is that? As a template. Next, here you are. The process of replication of DNA is repeated many times, and the segment or fragments of DNA can be amplified to approximately billion copies are made. So number of billion trillions copies are made. So the repeated amplification is achieved by the use of thermostable TAC DNA polymerase. So this is the very important TAC. TAC DNA polymerase. It is isolated from the thermophilic bacterium Thermus aquaticus. What is that? The TAC DNA polymerase is isolated from the what is that? Thermus aquaticus. What is that? Thermus aquaticus. And the next one, it remained active during the high temperature induced denaturation of double stranded DNA. So this is the very important. And the next one you are observing in a PCR structure. You are first in a PCR components. So DNA structure already we know. So double stranded structure we can cut the small pieces. So you are observing DNA sample and primase already you are nucleated adenine, thymine, guanine, cytosine, and these are the mixed the buffer solution and these fragments and PCR tube in the presence of what is the TAC polymerase enzyme. The PCR cycle can appear. The first process is denaturing and second process is annealing and the next one extension. So these three 95 degrees centigrade. That is the very important. And the next annealing means so 45 to actually 45 to 55 degrees centigrade. So primers for the formation of for the synthesis of new strands purpose you are observing primers. You are observing these are called as primers. So these are the opposite polarity they can form. So this side to this side these, and the next one template stand can form the this side to this side. So what is that? Primers bind the on the templates. So that process is called as what is that? Annealing. And the next one extension. These are extension primers are end to started to the and complete there the up to template stand area. So that time you are observing complete they can along with the template stand and completed the length of the equal template stand. So automatically they can form the new DNA. Means what is that? One stand can template can form the new synthesized stand. Means logging and leading stands automatically 1 2 2 4 4 8 8 16 16 32. These type of process we can get the within a short period we can get the number of billion copies by the PCR chain process. In this you remember these are the denaturing and annealing and extension. These are the very important, and you are concentrate the temperature is very important. So here denaturing is the what is that 95 degrees centigrade and annealing process is 55 degrees centigrade and extension process is what is that 72 degrees centigrade is very very important. So in this we can PCR reactions, we can use the what is that mixture of buffer solution, and the next important enzyme is what is the TAC polymerase. So that is a very important. TAC polymerase enzyme is isolated. So that is a very important. Isolated from the what is that a thermophilic bacterium, that is the Thermus aquaticus. This is a very very important. Thermus aquaticus is very important. And the next one you are observing PCR process you are observing. So this is the parent DNA, and the step one process is what is the denaturation. 95 degrees centigrade means 5 end to 3 end stand, it is the template stand. And the another one is the opposite polarity 3 end to 5 end stand. These both stands are both template can the separated you are observing. So these are the separated or cleavaged as a two template separate stand. These processes are called as what is the denaturation. So these process can occur in the specific temperature, in the presence of specific temperature. That is the what is that 95 degrees centigrade. That is the very important 95 degree centigrade. Then after annealing, annealing means which are separated the template, they can form out the opposite polarity a small fragment, they are called as what is that primers. So primers are here formed this small segment or fragment. So this side is what is that 5 end to 3 end, and next one this side 3 end to 5 end. So 3 end to 5 end and 5 end to 3 end is opposite polarity, they can form out the, so below the template stands. So and these processes occur in the specific temperature at 55 degrees centigrade. Then after these, during these formation is after during the primers is involved for the makeup the a new stand below the or above the template stands. That process is called as extension. So it can be observed in the presence of 72 degrees centigrade temperature. After these can be primers can form the new template stands. The new synthesized stands are also known as leading stand and lagging stands. That's why you are observing 5 end to 3 end below another stand is formed. So that's why here this is the one DNA, and the 3 end to 5 end above the one new synthesizer form is formed. So that's why you observe the what is that two new DNAs are formed. So these can two are formed the four are formed the eight are formed the what is that 16, 16, 32, 64. These like within a short period we can get the a number of billion trillion copies. So that is the very important. In this diagram you are also observed that very popular is what is the DNA polymerase enzyme, and the next one primer. So DNA primers tag DNA polymerase and nucleotides formation. So these are the very very important components for the process of the PCR. So here you are observing, so these three steps are very very important, understand. So one is the 95 degrees centigrade, 55 degrees centigrade, and the 72 degrees centigrade. So PCR steps already now we discussed. A single PCR amplification cycle involves three basic steps. That is the they are what is that denaturation, and the next one is what is that annealing, and the next one extension. So uh, extension, next one, first one is the denaturation. So already we know the denaturation is what is the DNA is heated to high temperature, usually 90 to 98 degrees centigrade or 95 degrees centigrade. In this denature means the DNA templates can be separated. So that process is called as denaturation. So exactly temperature you remember it here, don't confuse. 95 degrees centigrade is very important, and sometimes 98 degree also sometimes we can use it for the denaturation, but exactly you can remember it only what is the particular 95 degrees centigrade, don't confuse here. So next one, denaturation and annealing and extension. So here annealing is what is that, here 40 to 60 degrees centigrade, but you are exactly you remember it what is that 55 you remember. If 55 is not there remaining, that's time you take the answer another one. Okay, exactly, you can observe that 55 degrees centigrade. Okay, don't confuse. And the next one, here you are observing, so the two oligonucleotide primers analyzed to each of the single standard template DNA temperature usually 40 to 60 degree centigrade, at 75 is the very very important, 72 to 75 is very important. And extension, TAC DNA polymerase synthesis the DNA region between the primers and optimal temperature is 72 degrees centigrade. And the next, these PCR uses is, you are clearly observed that. So uses is diagnosis of pathogen and specific mutation, and DNA fingerprinting, uh, rental diagnosis, and gene therapy. These are the very very important for the detecting the crime also. So means what is that? That is the DNA fingerprinting. So here you are observing uses for the PCR in a medicine, detecting the infectious organisms and discovering the variations and mutations in genes, and the especially human genomes. So in a medicine purpose PCR uses. And the next one, the Human Genome Project in the DNA sequencing, and the law is genetic fingerprinting. And evolutionary biology uses is DNA analysis for taxonomic classification, and zoology purpose research on animal behavior, and ecology purpose studies of seed dispersal, and the reducing illegal trade in endangered species, and monitoring release of the genetically engineered organisms. So archaeology purpose and paleontology purpose, anesthetic DNA, and the analyzing the plants evolutionary purpose, and the plants habitat also we can habitat nature in the evolutionary, so that can be uses we can identify the with the help of the PCR. So mainly is what is that diagnosis of the pathogen and specific mutations is very very important. PCR is very use, you observe these points also. So now insertion of recombinant DNA into the host cell. So organisms, here you are observing, there are several methods of introducing the ligated DNA into recipient cells. So recipient cells, after making them competent to receive, take up the DNA present in its surrounding. RDNA can be forced into such cells by incubating the cells with the recombinant DNA ice. So this is the very important. Cells with the recombinant DNA ice, followed by the placing them briefly at 42 degrees centigrade. So that is important, 42 degrees centigrade, heat shocked, and then putting them back on ice. This enables the bacteria to take up the rDNA. So that is the very important for introducer rDNA into host cells, recipient cells. Some of the important methods are there. So one is the micro injection method, and mm, biologistic gene gun method. So in this micro injection method means directly give to the injection to the nucleus. So that is called as micro injection method. The nucleus direct injection method. So these are called as micro injection method. In this method, RDN is directly injected into the nucleus of an animal cells using capillary glass scopic pipette we are using. So micro syringe method, these are also called. So means directly we can use it the injection like method to insert the nucleus. So that's why it is called as micro syringe. So gene gun method, in this method cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA. This method is suitable for plants. Important crop plants like wheat, rice, and maize have now been transformed by this method. So use this method for the pathogen vectors, which when allowed to infect the cells, transfer the rDNA into the host. So here you are observing gene gun method. So here what is that? Plastic disc with the DNA coated gold particle. These are the, so directly pressure and the targeted to the disc stopped by the scanned DNA. You are observing. These are the target plant cells. Directly gun method using they can come fastly and enter into the plant cells. So this is the direct gun shoot gun method. So here you are observing directly, this is a gun like method. So directly leaves we can using this method. So here you are observing, this is the method. So simplest method of direct introduce of DNA into the target cells look like a pistol, but works more like a shotgun. Golden pellets, first described as a method of gene transfer into plants. Johnstown, Sanford at Cornell University in 1987. So anyhow, these are we can use it for the easy transforming to the host tissue. So these method can we can use it. So now you are observing some questions. Gel electrophoresis is used. Uh, what is that used to used for? So first one, construction of rDNA by joining with cloning vector. Next, isolation of DNA molecules, and next cutting of DNA into fragments. So separation of DNA fragments according to their size. So according to their size is very important. Separation of DNA fragments according to their size. Answer is D. So next, the term competent refers to increasing the competition between cells. Next, making cells impermeable for DNA. Next, increasing the efficiency with which DNA enters the bacterium through pores in its cell wall. So next one, making cells permeable for divalent cations. So, cation, sir. Here answer is what is that? Increasing the efficient with which DNA enter the bacteria through the pores. Pores. So this is the very important. Next, which of the following bacteria is used as a vector for plant genetic engineering? So in this agrobacterium tumefaciens, bacteriophages, and thermus aquaticus. Next, Pyrococcus furiosus. So here answer is what is that? Generally we can use the agrobacterium tumefaciensis. Next one, which of the following microbes transform normal plant and animal cells to cancerous cells respectively? So retrovirus and rhizobium, and Escherichia coli and agrobacterium tumefaciens. Next, agrobacterium tumefaciens and retrovirus, and the agrobacterium tumefaciens and rhizogenesis. So answer is agrobacterium tumefaciensis and the retrovirus. Next, read the given statements and select the correct option. So here statement one, the tumor inducing plasmid TI plasmid act as a cloning vector in recombinant DNA technology. Statement two, the TI plasmid which is used in the mechanism of delivering genes to a cell remains pathogen. So in these both statements one and two are correct. Statements one is correct, but statement two is incorrect. Next, statement one is incorrect, but statement two is correct. Next, both statement one and two are incorrect. So B is the statement one is correct and statement is incorrect. Your statement one is correct. So what is the tumor inducing TI plasmid? Next, what is that? The TI plasmid which is used in the mechanism of delivering genes to a cell remains pathogenic. So it is not correct. So only one statement is statement one is correct. Okay, next one. The correct sequence of making a cell compound competent is treatment with divalent cation, uh, cation, or incubation of cells with recombinant DNA on ice, heat shock 42 degrees centigrade placing on ice. Next, heat shock 42 degrees centigrade, incubation of cells with the recombinant DNA on ice, and treatment with the divalent cation, placing on ice. Next, treatment with the divalent cation, placing on ice, heat shock 42 degrees centigrade, incubation of cells with the recombinant DNA on ice, heat shock at 42 degrees centigrade, treatment with the divalent cation. So in this you are already so what is that? So here 42 first on the what is that? Placing on ice, B answer. So next, which of the following is required for microinjection method of gene transfer? Microparticle and the pipetters. Next, divalent cations, UV radiations. Answer is what is that? Micropipetters is the pipettes or for the what is that? Microinjection methods. Next, Process used for amplification or multiplication of DNA in DNA fingerprinting is polymerase chain reaction and the southern blotting, and next northern blotting, next none of these. So here answer is what is that polymerase chain reaction. Polymerase chain reaction, A is the answer. Next one, here you are observing in the isolation of DNA, the removal of protein and RNA is RNA is carried out by enzymes and respectively. So lysozyme, ribonuclease, next one protease and cellulase, and the next one what is that protease and ribonuclease, next one ribonuclease and chitinase. So in these you are observing, so what is that in the isolation of DNA removal protein, so what is the protein and RNA is there. So protein means proteases and RNA means the ribonucleases. So here during isolation of genetic material, the chemical used to precipitate out the purified DNA, so bromophenol blue, chilled ethanol, ethidium bromide, next both A and C. So answer is what is that chilled ethanol. What is that chilled ethanol? Next, match the column 1 and with the column 2 and select the correct answer from the given codes. So column 1 area, so column 2 area you are observing recombinant DNA technology. Next here chilled ethanol, so precipitation of DNA and the PCR. Next ethidium bromide. So already in this you are observing DNA staining, gene amplification, and the genetic engineering. So in this you are observing A, 4, B, C, D. So answer is A. So one is A4. You are observing. A4 means what is that? Recombinant DNA technology is what is that? Genetic engineering. So precipitation of DNA is ethanol. Already you know ethanol. So C is what is that? PCR. So gene amplification. Next one D, ethidium bromide. Ethidium bromide is what is that? D is so ethidium bromide is what is that? Staining. So that's why here answer is what is that? A. So clearly you are observing. So clear. So recombinant DNA technology, genetic engineering, precipitation of DNA for the chilled ethanol. PCR is what is that? Gene amplification, and ethidium bromide is what is that? DNA staining. Okay, next. Here you are observing. Match the column 1 enzymes with column 2 characteristic activity and select the correct answer from the given codes. So here you are observing tag DNA polymerase and exonucleases and the proteases and chitinases. So what is that? Already you know DNA polymerases. So polymerases is what is the use? Exonuclease is simple already we know. So simple the what is that? Chitinase. Once what is that? Chitinase the fungal. Proteases means protein. So degrade protein. So C is what is that? Simple. First you simple understand. So C is what is that? Two, and D is what is that? Degradation of protein. So that's why you are observing here. Answer is what is that? D. A3. A3 means what is that? Tag polymerase. Stable above the 90-degree. Next one, what is that? B1. So B is the what is that? Exonuclease. Cleavage the what is that? End of linear DNA. And the next one, what is that? C is 5. C is what is that? So proteases. What is that? Degradation of proteins. Next D. Chitinase. What is that? Breakdown of fungal cell wall. So this is the so column 1, column 2. 13th question is which of the following is not correctly matched for the organism and its cell wall degrading enzymes? So plant cell cellulase, and the next bacteria methylase, and fungi is the what is that? Chitinase, and the D is what is that? None of these. So already correct. Fungi is what is that? Chitinase. Plant cell is cellulase, but wrong answer is what is that? Bacteria is methylase. So that is the wrong answer. Next one, uh, in a polymerase chain reaction, temperature required for the steps, denaturation, annealing, extension are required. So in this, you are observing 94 degree, 40 degree, and 72 degrees given the what is that in a polymerase chain reaction temperature required for the steps. So answer is A. So this is the very important chapter for you understood and concentrate these type of questions. Okay, thank you all of you.